This lecture will be a brief introduction to some data structures and algorithms related to structural bioinformatics, in particular to protein structures. We're going to see a bit about the history and conventions of protein structure visualization, and then move on to the Protein Data Bank, where you can find data on protein structures. I'm going to talk a bit about the structure of the data formats of the files, and also one detail about how the biological units are stored in that data. And then we're going to look at algorithms for comparing different structures, in particular for comparing proteins. That will be the example I'll use, but this can be generalized to molecular structures in general. We're going to see why we should want to compare them, and different problems and different solutions for these problems, and also using the comparison of protein structures to align align the proteins with the structure instead of just using the sequence. So let's start with protein visualization, and one thing that it's easy for people to not know is that actually the problem of visualizing proteins precedes the creation of computers like those that we know now. When the first models were created of the structure of proteins, like insulin, for example, the visualization of those structures had to be done creating actual physical models of the protein with wires and plastic beads and so forth. So this is an example of the whale myoglobin model, one of the first proteins whose structure was determined, and some conventions for representing macromolecular structures in the computer came from those days when they had to be represented in this way without computers. So only in the late 1960s were there computers that could represent these images. So in this case, this is a cathode ray tube, and the lines there are drawn by shifting the electron beam across the tube. So it's actually drawing the lines in the screen, and this is a grandparent of the mouse that we can use to rotate the protein and see in different perspectives. And this would be a projection calculated by the computer and then drawn on the screen. Only in the 1990s these methods of generating images with rasterization, which is the electron beam is going through the image line by line and representing the different colors. This started to be used in common home computers. So this at this time, this Rasmol software appeared and popularized the representation of molecules, even for researchers without access to very powerful computers. This adapt the kind of conventions that people used before having computers. So these representations somewhat mimic what was being used before, with the plastic spheres and the connections and so forth. So some representations can be at a higher level, showing, for example, secondary structure. In these cartoons represent with these ribbons here in a helical shape the alpha helixes. They can also represent beta sheets with flattened ribbons like this. The convention for colors, they are called the CPK colors for these authors, Corey, Pauling, and Colton. They created these conventions of using black for carbon, red for oxygen, blue for nitrogen, white for hydrogen, and so forth. So this has been extended over the years, but there is some convention about the colors used for different elements. And even these conventions for visualizations, such as space filling, where each atom is being represented approximately with its van der Waals radius, or these ball and stick representations, where we have these sticks representing the connections between the atoms and small spheres representing the centers of the atoms. So note how these representations come from the original ways of physically creating a mock-up of the protein. So we had to actually glue things together in some way with wires or fixing the atoms to each other so that we could create a physical representation of the molecule. Of course, all of these representations are fiction, because the protein is much smaller than the wavelength of visible light. We would never be able to see proteins with our eyes, even if we magnified the image. And also these molecules are not static. The folding of small proteins can take milliseconds, so they are vibrating at speeds that are way beyond what we could see, even if we could amplify the image and look at it. So basically these visualizations represent average positions. We can represent mobility by showing families of different structures, and we can use colors and other attributes of the image to represent physical and chemical attributes of the molecule. So, or basically having just a nice image about some structure or some protein that we're interested in. So remember that these are conventional ways of representing the proteins, and it's basically a method of visualizing some properties or average positions or something like that. Okay, so more interesting for our purposes is how this data is stored and how we can access the information on protein structures. You can do this in the Protein Data Bank. This is a global collaboration of different labs, but it's basically a repository, the main repository for macromolecular structures. At present, there are nearly 200,000 structures deposited there. You can find there the atomic coordinates data. You can find many tools for searching, visualizing proteins, and even relations to other databases with genes and function and protein families, classification, and so on. The main information about the protein structure is stored in some files. I'm going to talk mostly about the original Protein Data Bank format file. That is a text file. You can open it with some visualization software, or you can even read it, open it with a text editor and look at it. It has general information about the literature references, the organism, the experimental techniques, and so on. And then it has this important part, which is the set of atomic coordinates for all the atoms in all the residues of the protein, additional information about secondary structure, the sequence, and so on. And also, I'm going to talk a bit about this, the relation between the structure that you have in the Protein Data Bank file and the actual biological unit of that protein in living organisms, because the data that is stored in the file may not correspond exactly to the biological unit. So let's look at the file itself. This would be the beginning of the file. There is this header which has a title and a name for the protein, references to where the structure was published, and the name of the molecule, and some data about the techniques used for determining the structure. Then you have details on the X-ray. For example, most of the structures are determined by X-ray diffraction. So here you have details on the X-ray, on the acquisition of the diffraction patterns for computing the structure. And then these are symmetry operations that you can do on the coordinates that you have there in the PDB file to obtain the unit cell of the crystal. So this is mostly interesting for crystallographers, not for actually using the structure. But then you have also this information about the biological unit. I'm going to talk about this a bit more further on, but you have this information about how to compute the biological unit of this protein from the coordinate data that you have in the file. And then you have information about the sequence of these proteins. In this case, we have two chains, A and B. So these are the sequences of chain A and chain B. Data on secondary structure. For example, we have beta sheets between these residues, and this can be used by visualization software to indicate where these secondary structure elements occur. And then we have the coordinates of the atom. So this is the format where you have a unique identifier for the atom, an identifier for the residue type here, the identifier of the chain, there can be several chains, the atom, and the coordinates here, x, y, and z. And then there can be additional information like occupation factors in the crystal, things like that, that can be here at the end. Or it can even include extra information. Some types of software can export these files with replacing this data with any indicator that we may want to see, things like charge or things like that we may want to visualize. But the main information is here, the coordinates and the identification of each atom. Note that there are no hydrogen atoms here indicated, and the reason for this is that hydrogen atoms can be added computationally. Once you have all the heavy atoms, it's easy to compute where the hydrogen atoms can be, because they have to be, we know how many hydrogen atoms are connected to each type of atom, so we just need to add them there. And also, in general, hydrogen atoms are not visible in X-ray crystallography. So the electron density is too low. They cannot be, the coordinates cannot be determined, and so they are left out of the PDB file, and then can be added computationally by just knowing how many hydrogen atoms we have on each of the heavy atoms. The PDB file also includes these heteroatoms, which are atoms belonging to anything that is not the amino acid residues in the case of proteins. So here we can have some iron atoms. In this case, this is an iron sulfur protein, so these are coordinated to the sulfurs in cysteines. And also these water molecules, so these are the positions of the oxygen atom of water molecules that are attached to the protein in the crystal. Usually the crystal has a lot of water molecules, and these may also be indicated in the PDB file. Then there are additional connections. The connections between atoms inside the residues are all put automatically by the software that reads this file, because they are always the same. But additional connections, for example, between these irons and cysteines, disulfide bridges in between two cysteines or things like that, these are all added extra here at the end of the PDB file. So this is how the PDB file is structured. For example, here we see the position of the iron atom and connected to the 4 sulfurs of 4 cysteines here. And these connections are included here at the end of the PDB file. There are other formats that you can use to download the protein structure. There is this XML format on the protein data bank. So basically what we have is these values, such as the X coordinate, the ID of the atom, and so on. They are all stored here in this XML format. So we have all these tags indicating what each field is. This may be not, at least personally, I find this a lot less practical than the original format, although this is a more modern format and has some other functionality. It's better to be used by computers to add different types of information without creating problems for parsing the original data and so on. So it's a lot more extensible, which is precisely the idea of XML. And this MMCF is also a classical format for structure. Some software uses it, but it's similar to the protein data bank, the original file format, but these tables are preceded by a list of what the columns mean, and then we have the actual table. So this is also this is based on this star format, self-defining text archive format, which had this convention of first indicating what the columns are before each table, so that the file itself has some indication about what it represents and what is its structure. In addition to the files, you can also see a lot of information about the protein structures if you visit the Protein Data Bank. So you can download these files from the Protein Data Bank, but you can visualize the structures on the summary page for each structure. You can see its membership to different classification. For example, SCOP is a database of classification of structures, and the protein family and things like that. You can also find similar structures by sequence similarity and see how in which regions they are similar, the results of this query, and so on. So basically, the Protein Data Bank gives you an interface to a network of databases and services that you can use and link from there to many different services. Okay, so it's not just a repository of structural data. It's actually something that brings together information from many different databases. Now, one important thing, if you're actually using the data, the structural data, to do something with the protein, is to understand that what you have in the PDB file is the asymmetric unit. This is the smallest set of coordinates from which you can, by rotation and translation, create the unit cell, which is then repeated by translation in the entire crystal. If you're doing crystallography, you have a very well packed and ordered crystal, where the proteins are packed always in the same blocks, in the same groups with the same orientations. And so this would be the entire crystal can be created by copying this unit cell and translating it in a regular lattice. However, the unit cell may be formed of these asymmetric units, of these smaller units, that can be copied and rotated around some points to create this cell. And we don't need to store the whole cell if this is the case. So we only need the coordinates of the asymmetric unit, and this is what you have by default in the PDB file. You have also the option of downloading the biological unit, but in the basic PDB file, you have this asymmetric unit. So what we need here is this information about the relation between the set of coordinates we have in the file and the actual shape of the biological unit. In some cases, for example, in this protein, they are all identified in the Protein Data Bank by these 4 alphanumerical codes. So these 2HHB for human deoxyhemoglobin. In this case, the asymmetric unit is the actual biological unit. It's this hemoglobin tetramer with 4 chains, and this is what is stored in the PDB file. However, for human oxyhemoglobin, we have in the PDB file only the dimer, so only 2 of the 4 chains, and then we have this information saying that we can build 2. So there are 2 parts here for our biological unit, and we have to apply these transformations to chain A and B. So basically, we keep them 1 copy exactly in the same place. These this matrix here that has 3 rows and 3 columns is just the identity matrix. This is the first transformation we need to apply, and we do not do any translation. So we have here a matrix for rotation and here a vector for translation. But the second operation that we need to do, also applying to chains A and B, is to do this rotation here. So you see it's 180 degrees on this axis and also on this axis. So basically what we are doing is we start from here and we have 2 copies. One that is the same in the same position, and the other that is rotated around the central point here over the 2 axes. So it creates the other part that is missing from the tetramer. And this is the actual biological unit. This is what we have on the PDB file. In some cases, we can have an asymmetric unit that has more than one biological unit. For example, in this protein file for goose hemoglobin, we have actually 8 chains in the asymmetric unit, which is in the PDB file, but the protein itself only has 4 chains. It's a tetramer. And so the information that we have here is that to obtain the first biomolecule, we apply this transformation to chains A, B, C, and D. So the first 4 chains, we apply the identity transformation. Note that there is no translation and no rotation. This is the identity matrix. And then to obtain biomolecule 2, we also apply this identity transformation, but to the other 4 chains. So basically, this is giving us the information that we have in the PDB file 2 different biological units, one composed of the first 4 chains and the other of the following 4 chains. So if you want to interpret the PDB file into a useful structure, either you have to take care to download the already processed file with the biological unit, or if you use the base PDB files, you need to pay attention to this information here about the transformations required to obtain the biological unit. Okay, so now let's look at one thing that we can do with protein structures or with macromolecular structures in general is to compare different structures. So structure is an important part of protein function because proteins are even selected for having some stable structures that can interact and work in some particular ways. If you were to generate random sequences of amino acid residues, it's very unlikely that this would give any protein with a unique and stable structure. But the proteins that exist in living organisms were selected by evolution to have these stable structures. And so not only is structure very related to the function, how the protein interacts with other proteins, but it also tends to be more conserved than sequence. So you can have proteins with sequences that are very different, but still be very close to one another in structure, have a very similar structure. And this allows you to find more distant relations between the proteins, because the similarities in structure last a lot longer during evolution than homology in sequence. Also, we can be interested in seeing how proteins change conformation as they interact or as they act in some way during catalysis, for example, or other interactions. We can compare different conformations to have an idea of the mechanisms of how they act, and also we can see, using structure comparison, measure the variety within protein families. So look at which parts must be always in the same structure and which parts can vary more. It's also used for classifying and organizing proteins. So these CATH and SCOP schemes that you can find online, these databases organize proteins into different families, superfamilies, and so on, depending on both sequence and structure. So the problem that we have here is to try to find a way to measure how alike or different are the structures of two proteins. The question is whether we're going to measure this using all the atoms, using only the main chain, the backbone atoms, using just one atom per residue, or things like that. We can compare the coordinates, or we can take into account also properties, and we need to decide whether or not we're going to try to measure this over the whole protein, or if we're going, for example, to overlap some parts and measure how other parts move relative to each other, things like that. So there are lots of different applications of these structural comparisons, and exactly what we want may depend on the application that we have. I'm going to talk a bit about some differences here as in the context of Superpose, which is a software that you can use online if you want. Now, two basic approaches algorithmically to solve this problem is either to first try to overlap the two structures as best as possible and then measure what difference remains, or we can look at geometric relations within the proteins, such as distances, angles, and so on, and compare these with the other structure. But in any case, we need to have some way of scoring how different the two proteins are, and we also need to search the best ways, which parts we need to compare with each part, or how to position the two structures, in this case the two proteins. So basically this is what these algorithms have in common. They need some way to score and some way to optimize the score or search for the best matches. So let's see one very common score, which is root mean square deviation, and this is basically we are summing for all the atoms the square of the distance between the atom in protein V and the corresponding atom in protein W. And we're going to sum these squares for all the atoms, take the average, and then use compute the square root. So this is the square root of the average of the square differences, or the root mean square deviation. The reason for doing this intuitively is that if we were just adding the distances, suppose that we have this circle corresponds to this circle and this triangle to this triangle, and we want to measure the difference between these two structures, the orange and the blue. And if we would simply sum the distance, then there would be many different possibilities which would give us the same sum. So whether we matched the triangle and approached the triangle to the triangle and left a larger distance between the circles, or vice versa, we would have the same sum of whatever was left over here in the distance. If we use a root mean square deviation, we have this quadratic function that penalizes larger values more. So this will give us the optimal solution, would be one that would try to balance out all of these distances so that we avoid having large deviations. So instead of having some large deviations here and small deviations here, they would even out and we would have a better match, a better superposition of the two sequences. So this is just an intuitive justification. A more formal justification is that this minimization of quadratic errors corresponds to a maximum likelihood solution when we have normally distributed error. So we saw this in linear regression some lectures ago, and it's the same idea here. If we assume that there is some normal distributed random noise between the two structures, then minimizing the quadratic error will give us the maximum likelihood solution. So what we need here to compute the root mean square deviation is to find the match between each atom in one of the structures, in this case V, and the corresponding atom in the other structure. Once we have this, we can measure these distances, but note that we don't want to measure these distances with V and W placed randomly somewhere in space, because otherwise we are just measuring mostly the distance between the two proteins. So we also need to search for the best overlap between the two so that they are optimally placed, in order that the distance that we're measuring here is only the residual distance after we approach as much as possible the pairing atoms together. So let's see an example. We have these two proteins, one represented in blue, another represented in orange here, and they have a similar sequence, and we want to measure how different the structures are. If we look at them like this, the structures seem to be very different, but they are not correctly superimposed one to the other. So first what we need is to look at the sequence, align the sequences, and on all the matches that we have between the sequences, we are going to make a correspondence between, for example, the alpha carbon of the residue in the blue protein and the corresponding alpha carbon in the matching residue in the orange protein. So we can do this after we do a sequence alignment. Note that some parts do not align well. There are some gaps here, but in general we have an alignment over most of the proteins. And then what we do is we rotate one relative to the other and translate it. in order to minimize the root mean square deviation measured over all of these matching pairs of atoms. And this is what gives us the best superposition of the two proteins, and we can visually now get a better idea of how the proteins differ in structure, and also now the root mean square deviation measured in this particular orientation gives us that residual difference between the two when they are overlapped as best as possible. So this would be actually the measure of difference, of structural difference between the two proteins. We can do this in different simulations, in different situations, So we can have the exact same molecule in different conformations, and in this case, it is trivial to match the atoms, because if it's the same molecule in different conformations, we match each atom to that same atom in the other conformation. If we have proteins with similar sequence, we can do like in this example, we do a sequence alignment first, and we use those matches to compute the root mean square deviation. If we have molecules that are partially similar in the sequence, with a good similarity in some part, we can do the overlap there and ignore the rest, for example, because at some point, if the sequences are very different, we cannot find the correct matches and measure these structural distances. In the case of having very distant sequences, then we may no longer be able to use sequence alignment to superimpose the two, and we may have to move on to structural alignment. So I'm going to talk a bit about that also in a few slides. In the case of structural alignment, we can also, instead of doing this particular way of finding the match first, because generally we are going to use structural alignment when we cannot match the sequences, then what we need is to try to match the sequences as best as possible based on the structure. So we note that we have here a bit of a problem when we're doing structural alignment, because we need to compute the structural differences between the proteins for this alignment, but we cannot match the correct atoms from one protein to the other based only on the sequence. Usually structural alignment is done when we cannot do the alignment on the sequence. So we have to do those kinds of approaches that we saw previously in different contexts like clustering and so on, where basically we start with an initial guess, and then we iteratively improve this guess by going back and forth and recomputing this alignment. So one way of doing this using distance matrices is to begin with a tentative match between the two proteins, and then compute the intermolecular distance between all the pairs of possible pairs of reference atoms, looking at, for example, the alpha carbon of atoms in one of residues in one protein to the alpha carbon of residues on the other. We can compute this matrix with all the pairs, but first we have to superpose the two structures as best as possible so that these distances represent an actual difference in the structures. But this superposition requires this initial tentative alignment that tries to match parts of the two structures. After we do this, we can, when we have these distances, we can check the similarity between the different parts of the molecule by looking at the pairwise distances in the matrix, and then use this to refine the tentative alignment that we had before and recompute this. So basically this can be done with these matrices with distances between atoms of one protein and all atoms of the other. Usually not all atoms are used, but only one for each residue, for example, the alpha carbon. We can also do this by looking at patterns of distances inside each protein. So in this case, we are going to create matrices of distances between all pairs of atoms in one protein, and then the same thing in the other, and try to match sets of distances in these matrices to find those substructures in the proteins that are more similar between the two. So basically, from these distance matrices, we can compute differences between different parts of the proteins, use these graphs to show us which parts we need to overlap more. We can also use descriptions of the neighborhood around each atom by computing vectors to atoms in the neighborhood on different residues. I'm going to talk a bit about this in one of the algorithms for aligning the structures. So let's see first this part of superposition that we saw for the computing the root mean square deviation. So basically, we can compute this RMSD for any relative position of the two proteins, because we have the coordinates of the atoms for protein V and for protein W. And if we can match corresponding atoms between the two, we can compute this for any relative position of them. But now we need to find the rotation matrix here after we center both proteins on the same point. So we are translating them, so to overlap them on translation. All we have to do is to rotate one relative to the other, and basically we want to minimize this, minimize this quadratic error by adjusting this rotation matrix, so that after rotation the coordinates of the one protein are as close as possible to the coordinates of the other. This can be solved analytically, it has an analytic solution. If you want, you can see this Structural Bioinformatics book by Burkowski, but we don't need to go into those details. Basically, we can just solve this problem and find the matrix that matches as best as possible the coordinates of one protein to the other. So this part is easy to solve if we can make these pairs of corresponding atoms between one protein and the other. If you want to use this on a web server online, you have this Superpose server that computes these superpositions but to compare the different structures. So it can automatically compute the sequence alignment and then does this translation and rotation to superpose the proteins. If the sequences are identical, then it doesn't even need to compute the alignment, and it just does the superposition. But there are some situations, for example, we have identical sequences, but the structures are very different. For example, the protein has some hinge that can bend, and there are large conformational changes. So in this case, Superpose automatically identifies the common structures that are rigid and between which we have this kind of movement like hinges and things like that. So what it does is compute the distance matrices between all the alpha carbons and then checks which parts of these matrices are very different between the different conformations that we are presented. And then it will use the largest group as an anchor, superimpose that one, and leave the rest that moves more widely in different positions, so that we can better visualize the conformational changes. If there is a good sequence similarity, then it does sequence alignment first and minimizes the RMSD. If the sequences are very dissimilar, then it tries to align using secondary structure. So instead of using the actual sequence of amino acid residues, it looks at the sequence of secondary structure along the protein chain and uses that to align. But in this case, the alignment may be rather poor if the sequences are very different. So if the sequences are really different, we may want to align the proteins not based on the sequence itself, but on the structures. And so here we can look at those matrices that we saw, that we can also use to measure dissimilarity between the structures, or we can look at the structural properties in the neighbor of each amino acid residue. The reasons for doing structural alignment is that if sequence similarity is low, there may still be good structural similarity because structure is more conserved over time. However, there is an additional risk that sometimes structure is affected by convergent evolution. There are some limitations on the kinds of structures proteins can have, and it can happen that two very dissimilar proteins end up having some structural elements in common because of convergent evolution. But one example of doing structural alignment with matrices, like we mentioned before, is to use these distance matrices that represent the distances within each protein. So we can have here the distances between alpha carbons on all along the chain, and this is represented in the vertical and the horizontal is the same chain here, and we are looking at the distances between these alpha carbons along the chain. And now with this kind of distances we can represent each protein with its own distance matrix and then look for patterns in subsets of these matrices that are similar to between the two proteins. So these indicate sets of atoms that have a similar network of distances, so these structures should be similar, these local structures. And to do this, each structure is broken down into virtual fragments. It considers 6 residues at a time and starts matching these groups of 6 residues between one protein and the other and find contiguous regions of residues that have a similar structure. So in this way, we can find ways of matching the two proteins for aligning them using the structure. Another way that I mentioned previously, that idea of looking at the neighborhood, spatial neighborhood of a residue, we can, looking at a particular residue, build a set of vectors. In this case, SSAP, this algorithm, uses vectors between the beta carbons of the different residues. The reason for using the beta carbons instead of the alpha carbons is that this gives us some information about the direction in which the side chain is, because the alpha carbon is on the main chain, but then the beta carbon is the first one on the side chain. Of course, we cannot use glycine in this case because they don't have these beta carbons, but for the other residues we can do this. And so for each residue we have a set of vectors that points to the beta carbons of the neighbors in the protein. And we can see this star, this three-dimensional star, as representing the local structure around each vector. And now we can do an alignment of to align both the sequences and these sets of vectors. So basically this is a double dynamic programming alignment where for 2 residues we try to align the vectors to see how well they can match, and then we use those matches to align the residues in the sequence. And basically we are aligning the 2 sequences using the vectors describing the structural similarities or dissimilarities between those residues instead of just looking at what the residues are. And this is how we can do the alignment between the 2 protein sequences considering the structure around each residue. Okay, so to sum up, we saw briefly some basic ideas about the visualization of proteins and how this data is stored in the protein data bank. And then we saw the problem and motivation for comparing structures in which we have these superposition methods where we first rotate and translate one protein to superpose with the other and then compute the residual distance between the proteins, or these that look at geometrical relations between distances, for example, distance patterns between the atoms and so on, and use that as a way to measure differences between proteins, and also for structural alignment. Structural alignment is important because structure tends to be more conserved than sequence. So even if you have proteins that are very different in the sequence, it may happen that you can align them and superpose them well, looking at the structure itself. So in this case, we can also use these distance matrices. We can use vectors to describe the local structure around each residue, and then use that for the alignment.